Okay, why we picked up there, but we are now going to move on to the very last part of the electron transport chain, which is the ATP synthase complex. It's technically part of oxidative phosphorylation and not really part of the electron transport chain. At one time, they called it complex 5 of the electron transport chain, but now this is why we have a new name that is oxidative phosphorylation. So, but if you think about it, regardless of how you classify it, it is one of the most essential protein machines in all of our cells. So we're going to talk about it in terms of its arrangement of polypeptides and how the conformational change in this protein and in these polypeptides result from that change of the protons from the intermembrane space, inner mitochondrial space down into the inner matrix. So we're going to talk about how that proton motive force controls ATP synthesis. And it's really, really, really cool. So the ATP synthase complex is composed of two structural components. And so we call it the F1, which is down here. This is F1, so all of that stuff down there. And the two images here are the bacterial ATP synthase complex and the yeast, which we use as a model of the eukaryotic, which is extremely similar to what we see in humans. So if you look at the two, the prokaryotic is extremely similar. It lacks a little bit of the detail that the eukaryotic has, but each of the subunits is extremely highly homologous. So we know that this is a highly, highly conserved and therefore very old piece of machinery in the cell. It's very cool. Okay, so the F1 complex, so all the stuff down here in the ATP synthase complex, this contains the part of the ATP synthase complex that has catalytic activity, that can actually do things. Then you have the F naught or the F0. It really stands for an O. The O right there stands actually for oligomyosin. And if you remember oligomyosin, you don't have to memorize this, but oligomyosin is a poison that's specific for bacteria that will inhibit the F naught channel, so that part of the complex. And so that's why it was named that way. So anyway, okay, so our F naught complex is what's embedded inside of the membrane, right? Up here is our intermembrane space. And then what's embedded in the inner mitochondrial membrane is that F naught complex. And so what has to happen? Well, we know that, let me kind of erase a little bit of this junk that I've drawn. We know that we have protons that are here in the inner mitochondrial space. And so we know that those protons built up because our electron transport chain pumped them into that space. And so we have a high concentration of protons inside that inner membrane space. And so the protons need to be able to cross the inner mitochondrial membrane, but they can't just diffuse. So they're going to have to pass through this complex. So the part of the complex that allows the protons to flow through is the F naught complex. And it's actually kind of complicated, and we'll talk about how that works shortly. So this is kind of structurally what we talk about. But you have to also be able to look at the ATP synthase complex in terms of function. So we're going to look at the functional parts of the ATP synthase complex, and they're not necessarily separated into what's embedded in the membrane and what's not. So, and you'll be able to pause at different times in the video to write down all the extra stuff that I have. So just kind of follow along as I go and pause when you need to. So here are our three big functional parts of our ATP synthase complex. The first one is called the rotor. So way over here, this is the rotor. The rotor has 3 subunits. So our 3 subunits are our gamma, our sigma, and our epsilon. And what happens is that our rotor is going to rotate, right? So it's our 3 complexes and it rotates and the whole thing rotates. So each of these subunits, these 3 subunits, are going to rotate together and they're hooked on to these channels up here. And so what we'll see is the whole thing is going to rotate all together. And it's really, really cool how this actually works. So that's part of, you think of a rotor, it's something that is allowing movement to happen. And when is that movement going to occur? Only when protons either enter or exit, and this is the ring. So this C10 means there are 10 different pieces, 10 different polypeptides. And so that's the ring that once our protons enter and exit, that's what's going to induce the conformational changes, which allows that rotor to move. Our second piece is the headpiece. The headpiece is our catalytic. So whenever you think catalytic, here's our headpiece. Whenever you think catalytic is the enzyme. This is the mechanism, and this is ATP synthase. So what is the headpiece going to do? It is going to make ATP. So it's hexameric, meaning hex meaning 3, meaning 6. We have 3 alpha and 3 beta subunits, so that totals 6, so it's hexameric. And this is what's responsible for ATP synthesis. So there are technically 3 sites for ATP synthesis, and they're all in the beta subunits. So each of these 3 beta subunits, there's 1, there's 2, and there's another one, 3. So each of these beta subunits has the ability to take ADP, an inorganic phosphate, and create ATP. So what's important to kind of remember is as we're looking at this, these aren't separate in the cell. So this rotor, so if you see this rotor, this is the bulgy part of the top of the rotor. And this piece down here actually extends down the center of the headpiece. And so as that rotor moves, it actually induces conformational changes, which results in ATP synthesis. And we're going to talk about how that works in just a second. So the last piece is called the stator. And the stator has kind of 2 functions. And one of them is that it has 2 half channels for those protons to move. So really what happens, and I haven't said this before, so here's our stator right here, and our proton is right here, and he's going to flow in. And this is half of the channel. And you say, okay, well, where does he go? Well, he's going to go kind of at a turn, and he's going to attach to one of these C subunits. And once it makes a whole revolution, once this proton goes all the way around, then it's going to come out the other half channel. So the stator has 2 half channels. It comes into the first half channel, then it gets picked up by the ring. And then once it goes all the way around, it gets dropped off at the second half channel and can go into the matrix. So now this is matrix, and this is our inner mitochondrial space. Okay, so the second function of the stator is, if you think about it, stationary, right? It keeps our alpha 3 and beta 3 from moving when the rotor moves. So it hooks around and attaches to prevent these pink and green alpha and beta subunits from turning when the rotor turns. Because what we want is the rotor to kind of bump around inside that headpiece and induce conformational changes. And if every time the rotor would move, the headpiece would move, nothing would ever change. And so it wouldn't work. So this is a really, really good point to pause this video, go to the Norton site and watch this ATP synthase animation. It is phenomenal. Please, 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 please go and watch it. Okay, so now I'm kind of assuming that you have watched that ATP synthase video. So now what we're going to do is we're going to talk about these conformational changes that happen in the ATP synthase complex. We know that the protons are going to be in that inner membrane space, and they're going to need to flow through the F naught component. So it's going to have to go through those half channels. So, and in the video, it's the gray one there. But it's also gray here. So this is kind of what whoa, that was crazy. This is kind of what we're talking about right there. Okay, so the protons are going to flow through the F naught component. The F1 subunit, so that's the part that's actually inside the mitochondrial matrix, is going to undergo a conformational change. And it starts with that rotor moving, and the rotor bumps along and hits those alpha and beta subunits and induces conformational changes. So there are 3 different conformations that F1 subunit can be in. That is loose, tight, and open. And so if you think about this, why are there 3 different conformations? Well, each one of the times that rotor moves, it moves a whole 120 degrees. So if you think about that, right, 120 times 3 gives me 360. So now I'm back to where I was. So I can do it all over again, which is really, really important in biochemistry. So the whole point of that F1 subunit is to take the ADP and PI, bind them together to make ATP. And once that rotor combines the 2 and hits the open position, then the ATP can be released. So what results, what is the initiator for creating ATP is the protons entering our ATP synthase complex. What releases the ATP is basically the loss of that proton gradient, or the dissipation of the proton gradient. And that's what releases our ATP. So I want to go into just a little bit more detail, just so that you kind of have some really nice written notes that explain how this works. So there's 3 really core principles of this mechanism. And this is really important. So when we talk about the subunits, so this orange subunit right here, right, we call that the rotor. That's it's part of the rotor. And it contacts all 3 of the beta subunits, but it doesn't fit perfectly down the center. So if you kind of look, and it's really nicely shown in the animation, but if you look, right, whoa, there's a curve to it. So it can't have the same amount of contact with all of the beta subunits. If it were a straight stick down, it would, but it's not, it's curved. So it is contacting all 3 of the beta subunits, but each interaction between that alpha subunit and the beta subunits are distinct. So there are 3 different conformations that beta subunit can be in. And that corresponds to ATP binding affinity. So if the conformation, and these are all beta conformations, if the beta conformation is in L, it's going to bind ADP and inorganic phosphate. If the beta conformation is in tight, it contains the ATP, but the ATP is still bound. That's why they call it tight. Then open is when the ATP is released. So let's take one step back and think about our protons. So our protons are flowing through that F naught. And as the protons flow through the F naught, the subunit is rotating. And it rotates, if you look like up from the matrix, it's rotating counterclockwise. And it's really nice in the animation. It's hard for me to show you that, but it's really nice in the animation. So for every 3 protons that are in the inner membrane space that transition to the matrix, so now we're in the matrix, we get that conformational change. That conformational change is 120-degree rotation. So if we look at just one beta subunit in 120 degrees, that first 120-degree turn is going to go from the loose configuration to the tight. Then we go another 120 degrees, we go from tight to open. And then another 120 degrees, oh, look at this, we go from open to loose. So we started in loose, we ended in loose. And in that process, we're going to generate 1 ATP. And we have 3 120-degree rotations, which leaves us at 360. So we're starting back at the beginning. But here's the deal. How many beta subunits do we have? We actually have 3 beta subunits. So because there are 3 beta subunits, I should make 3 ATP from 9 protons going from the inner membrane space into the matrix. But it doesn't quite work like that. Of course, right? There's always got to be a caveat. It's not perfect. The rotation's not perfect. So a third of the time, you're going to need a 4th proton to make that 120-degree rotation. So actually what happens is when you have 10 protons moving from the inner membrane space into the matrix, then you're going to make 3 ATP. So, and we're going to see how that math works out really nicely in class when I finally see y'all. Okay, so now we're going to look at the same mechanism, but we're looking at it from a different angle. So now we're looking up at it as if we're standing in the matrix and looking up, right? Okay, so if you look at this arm, what you can see, this is our gamma subunit, right? That's our gamma subunit. And what we can see in our gamma subunit is that it's not perfect, right? It's not just a stick down the middle. You have these different ways that it's interacting with the beta subunits. And this is what induces that conformational change. So what I'd like to do is I'd like to follow one subunit all the way around. So I'm going to start with my configuration in exactly the same way I started here. I'm going to start on loose. Okay, so here I am. I have a beta subunit and I'm in the loose configuration. That's the way that the gamma subunit is bumping into the beta subunit. So right here, I am going to have that rotor turn 120 degrees. So this rotor is going to turn 120 degrees. So it's going to go in this direction. So what was this bump here is going to now be interacting with beta 2. So the bump that was with beta 3 is now with beta 2. So I had ADP and my inorganic phosphate bound. And so when I make that 120-degree rotation due to the change of 3 protons from my inner membrane space to the mitochondria, I am going to result in the formation of ATP, but it's not released yet, right? So we're kind of like monitoring just this one. So it's bound to ATP, but it's not released. In order to release it, I need 3 more protons to translocate. When those 3 additional protons translocate, then this rotor is going to move again. And now I have this open interaction. And so it's going to result in the 3rd configuration where ATP can be released and ADP-PI can come in. And so this is how we get 1 ATP per beta subunit for our 10 protons, but those 10 protons, right, are not just affecting one of the beta subunits, they're affecting 3 beta subunits, which is why we get 3 ATP instead of just 1. So this is a really nice little experiment that shows that ATP synthase really is a molecular motor. I really love this experiment. It's really cool. And so what they did is they took the ATP synthase complex and they removed all of this stuff on top, right? They got rid of it. And they took the rotor and they attached the rotor to an actin filament. And they actually they don't show you this, but they put a magnet on the end of this actin filament. And so what we can do is we can use this actin filament with a magnet. And you can put another magnet, right? And you can crank this ATP synthase molecule, right? And so depending on which way you would go, it results in either ATP formation or ATP hydrolysis. It was really cool. It's a really, really cool thing. Now, you can monitor how quickly this happens and that kind of stuff. But what I really want you to think about is how quickly is this happening in the cell, right? This is just an experiment, and it depends on how quickly we move the magnet, how fast ATP will be either synthesized or hydrolyzed. But in the cell, this is doing about 130 revolutions per second. Yo, that's crazy. That's really, really, really fast. Really fast. All right. So that is the end of section 3. So this is a good point. Now that you made it through, probably go back and watch that animation one more time. It sure can't hurt. All right. Have a great day.